Alrighty guys, today we are going to go through and do our notes for active transport. This is the final section of chapter 5, and I'll show you at the end in Canvas where you will need to go and submit all of these notes. Remember, you're not writing down everything, you're just writing down the stuff that is colored in red. So right here is where you're going to start. Sometimes a cell needs to pump materials against the concentration gradient, and that is active transport. This means that the solute is going from low to a high concentration. Energy in the form of ATP is needed. So here in passive transport, I have a guy and he's rolling a boulder down a hill and he says, wow, this is really easy. You don't have to exert any energy, he just lets it go and it happens on its own. On the other end, here's active transport, and this guy is pushing it up the hill. It requires a lot of energy, it's a lot of hard work, so that's active transport. Some examples of this are getting rid of waste in cells and moving nutrients into and out of the cell. Active transport moves nutrients, sodium and potassium, by a sodium-potassium pump. Animals want a higher concentration of sodium outside the cell and a higher concentration of potassium inside the cell. This means that sodium will leave the cell and potassium will enter the cell. So here's our sodium-potassium pump that I showed you when we did our vocab, and it's in a 3:2 ratio. We have 3 sodiums leaving. You add energy and 2 potassiums are going to come in. One of the phosphates is going to be taken off, and now you have adenosine diphosphate. The picture shows that each molecule of ATP can transport 3 sodiums out and 2 potassiums into the cell. Now, carrier and channel proteins not only assist with passive transport, but they also help with active transport as well. These proteins are sometimes called cell membrane pumps because they move substances from a lower to a higher concentration. So here we are, we're moving these little green particles from a low concentration to a high concentration. Here's an example of moving the potassium. Normally there's less potassium outside of your cell, but your cell likes to have potassium inside of it. So there is a higher concentration in your cell, and it's going to be adding more to it. Your cell is going to use something called a vesicle within your cell membrane. So some substances, such as macromolecules, are too big to pass through the cell membrane, even with a protein. These substances will use 2 methods called endocytosis and exocytosis. Both endocytosis and exocytosis require energy in the form of ATP. By definition, endocytosis is the process by which cells ingest external fluid, macromolecules, large particles, or other cells. Endocytosis takes in substances by them becoming enclosed by a portion of the cell membrane. The cell membrane is going to fold around the substance and finally pinch off into the cell. A vesicle is going to form from the pouch that pinches off from the cell membrane. So here you can see 4 little vesicles, and they have some type of a solute inside of them. We have our solute particles starting to come into the cell. You can see the vesicle is beginning to form. It's starting to pinch off, and in it comes. Penocytosis literally means cell drinking, and it's going to transport solutes or fluids. Phagocytosis literally means cell eating, and we're going to transport large particles or whole cells. Many unicellular organisms feed by phagocytosis. Certain animal cells are going to ingest bacteria and viruses that invade the body, and we call these phagocytes, and then they can be digested by lysosomes. Exocytosis is the process by which a substance is released from the cell through a vesicle that transports the substance to the cell surface and then fuses with the cell membrane to let the substance out of the cell. Cells are going to use exocytosis to release proteins, waste products, and toxins from the cell. This is how newly finished proteins are going to leave the cell after their journey to the Golgi apparatus. And here you can see the opposite of endocytosis occurring. You have your vesicle in the cell. The vesicle is going to become part of that cell membrane, and it's going to release those solutes. And that is it for active transport. So if we go over to our Canvas here, we'll see on Monday, sorry about that. It's Chapter 5 Notes Turn-In. So this is going to be all 4 parts of your notes. You're going to click on this assignment here, and you're going to turn them in. You can either upload them through a Google Doc, or you can just copy and paste everything into the text box. Alrighty, guys, that is it for today. Now, tomorrow we're going to do a couple review assignments just to start preparing us for our test.